So, good day everyone. My name is Seun Olufemi, and today I'll be speaking on bioinformatics designing and molecular modeling of mRNA vaccine against circulating variants of SARS-CoV-2 virus. Okay, so I appreciate the reviewing committee of the Beyond Science Initiative for giving me this privilege to present this research work at the 8th International Conference. So, as we all know that SARS-CoV-2 virus is the major causative agent of the disease infection known as COVID-19. And during the 2020, we had the pandemic, which led to the world lockdown. And as of January 30, the World Health Organization had made a declaration on SARS-CoV-2 and declared it as a public emergence of global health. And since that time, biotechnology companies have utilized various technologies to develop vaccine candidates against the virus. So, with the available vaccines now, there have been recent fallout, different publications, events, claiming that there have been fallout in the actions of the available vaccine due to majorly what we call inexistence of global inequity challenge and global inequity challenge, which is majorly as a result of two things. One, that most of the data that were used in the preparation of the vaccine came from preliminary data during the pandemic period, and two, there is an urgent need for there should be a vaccine during the period. So this has happened, and those vaccines on the later part were now being developed, now starts having fallout. So it was with these that we sought to develop and model an mRNA vaccine against the circulating virus of SARS-CoV-2 that would encapsulate all the circulating variants. And during this work, we covered about the major variants, which are the 5 major variants, and about 21 sublineages of the SARS-CoV-2 virus. So with this work, one of the things that we were able to do is that while trying to retrieve the sequence, we retrieve representatives of each variant and each sublineages across all the continents to show that there will be representations of each of these viruses across the continents. Then after we map out our sequence, we do antigenicity testing. Then therefore we try to predict our HTL using the IEBD tool. We predict our CTL using the net CTL. Then for our immunoglobulins with using the BC-PRED and ABC-PRED. Then before we eventually model finally construct our vaccine. So, and before that, one of the things we do is that since the major problem of mRNA vaccine is that the major fear of the people is that probably they might have been insertional mutagenesis, so we decide to blast each of our epitopes against the human genome to see that if there were regions which could easily lead to that, and we discovered that there was none. So we designed the vaccine, check for the physicochemical properties, the antigenicity, the allergenicity, the toxicity. Then also, after modeling these vaccines, we do several post-vaccine construct analysis to ensure the viability and the antigenicity of the construct. We do molecular dynamics so that we'll be able to check the fluctuation, how the particular vaccine construct will act under stress in the human body. We dopped it to see its affinity with TLRs. We check for its physicochemical properties, the instability index, the molecular weights, and we also check via artificial intelligence to also see the numbers of the immune responses, or how lengthy the immune response will be. So these are results. The results of our vaccine shows that in totality, our vaccine could cover about 68% of the entire world population, with over 75% coming from Europe, 69% coming from North America, 53% coming from Africa, and a whole lot of places, because we while trying to do the work, we try to use a reference set of analysts that has been implicated to cover across different populations, different ancestry in Africa, and this also shows the 3D structure of our vaccine. And our vaccine, while trying to make the primary structure, we imbibe the use of co-translational modifications and adjuvants to also increase the immunogenicity. Now, while evaluating the structure, we discovered that it has about 2,540 amino acid residues, about 285 kilodalton. The theoretical PI is about 9.20. The instability index around 48. Alpha index is about 65.85. And the Ramachandra plot here also shows us that our vaccine would probably the positioning of the amino acid in our vaccine, mostly about more than 95 to 98% of it occurs in the favorable region. So with our vaccine, this is our vaccine toll-like receptor complex. The two complex binds together, showing the interactions between the two boats, while the vaccine with toll-like receptor 9, also we also have a quantifiable amount of interactions between the two. Now, here is our immune simulation curve graph, which shows the level of IgM and IgG, which shows the level of our IgM, IG1 plus IG2 and IG2, then our IG. So it shows with this particular curve, we are able to determine the positioning that within the number of 0 to 5 days, that our Ag could rise to about, when injected between 0 to 5 days, could rise to around 75,000 thereabout, while between our IgM and IgG, which is very, very important, could also rise to between 0 to 15 days with around 700,000, with around 700,000 antigen per count meter. So our result shows that our vaccine construct is quite promising. Now, the population coverage of the construct, as I've earlier discussed, is about 68.897% of the entire world population, and then the approach we used was similar with the one with Ahmad and Lera 2020, and we also account that in our results, quite a number of sequences comes from Africa, and this is one of the approach we put in place to ensure that there is the problem of inequality in African representation is solved. So cross-reactivity of the selected episode was also screened, which also ascertained its potential safety risks from insertional mutagenesis. Then, based on our physiochemical properties, we are able to judge that our vaccine construct is going to be thermally stable, and then the half-life also is very, very interesting, because we need the half-life to predict whether it's going to induce enough immune response before it degrades. So due to the ever-mutating character of the SARS-CoV-2, the study that we have done has designed and modeled a potential vaccine candidate integrating the circulating variants and about 21 SARS-CoV-2 lineages, alright, which computational techniques as ascertained to be antigenic, non-toxic, and non-allergic, which under further validations can be validated via in vivo and in vitro studies. So here are my selected references. So in case you have any questions, you could reach out to me on mail via oluwaseunjr1@gmail.com, on LinkedIn at Seun Olufemi, on Twitter at Seun Olufemi9. And my acknowledgements go to the Beyond Science Initiative for this invite, Alex Baldwin Institute. Alex Baldwin Institute has been on hold, so that's the institute where a bunch of this work is carried out. And my primary institution, Ladoke Akintala University of Technology, Ogbomoso. I also appreciate my team members and the principal investigator of this institute, Dr. Elijah Kolawole Oladiopo, and I appreciate all the participants for your audience and your rapt attention. Thank you for listening.